asking, like, where are we going to, and I'm curious too, um, where are we collecting those rocks?
Okay, very good question. So when we map a site for the first time and see what its shape is and its topography, we try to guess where we might be able to find the best rocks. And by the best rocks, what we're talking about is rocks that have not been too altered over time, because we believe this seamount is really, really old. We think it's probably on the order of 70 to 80 million years old or more. And that's a lot of time for changes to be made with the rocks themselves. And so what we try to do is we try to find a spot where we might be able to find rocks that are the least altered, because they're usually the ones that are the best for aging.
Thank you so much.
So from some of the experiences that I've had in the past doing a fair amount of ROV work and submersible work, we found that these ridges that are extending out from the seamounts generally have very, very good rocks in addition to biology. And the reason being is a lot of the sides of the ridges have slumped away because these are so old. It's like anything up on land. You have a landslide that comes down. Well, these landslides happen underwater as well. And so you have a lot of the slides on the softer material that just slide away over time. But these ridges don't. And the reason is, we think, is because they can often have very, very dense rock that might have cooled inside the lava tubes rather than erupting out into the water. And so we look at these ridges, and more times than not, we found very, very good rocks to sample. So that's why we picked it. And when we get down there, though, a rock is not a rock is not a rock. And so you're going to see lots of rocks, but we aren't going to grab the first one that we see. We're going to be very picky and very choosy in which rocks we want to get. And so we're going to look.
Who's picky? Who's going to be the picky person and the choosy
Well,
person?
I've done this a lot, so I'll be very picky. And then we've got some geologists on board, a number of them. And if they don't think I'm picking the right rocks, they're going to let me know about it, and they'll be up here, and they'll be very picky too.
Oh, okay.
But rocks that generally are really good for dating are often what we call pillow lavas. And these are lavas, they have gotten out onto the surface, but in any case, you can often get good type of material from them. And also other types of rocks that's called either dike rock or columnar basalt, which we also hope to see. Another consideration is a lot of these rocks are glued together by something that's called manganese crust. And even if we see this beautiful, lovely rock that we want to grab, if we can't free it from the substrate or from the bottom, it's no good. So sometimes you might see us sort of poking a rock to see if it's loose enough for us to grab. So those are the types of considerations, and there's a lot of us who've had experience playing this game before, and I think with all the collective experience, we should be able to get some very, very good rocks and figure out when the seamount formed and how old it really is.
Yeah, that sounds very exciting. Well, I'm very interested in um, knowing what microbes are on those rocks too.
That's correct. And also a very good point, Data, because for Dr. Orcutt here, she's going to be looking at a slightly different kind of rock. She's looking for a rock that's not quite as dense as we would pick for aging. She's going to be looking for a little bit of a softer rock that's got some pores and things where microbes and bacteria and other types of things can live inside the rock itself. And we've got a couple of special boxes all sort of slotted for her in the front of the ROV for her samples that are going to be very tightly closed when we collect them. And they're going to preserve these microbes, and it's going to preserve the temperature if the ROV comes up. And so we're going to get a couple of those type of rocks, and then we're going to be collecting these denser rocks for aging too. So, and we're hoping for her guidance as well up here on what's going to make a good rock for her work.
Yeah, then we learned from the presentation yesterday that there are good rocks and there are bad rocks, but that that impression is depending upon the viewer, of course. The
Yeah,
That's right.
those that
Yeah.
are
yeah,
good rocks for dating aren't necessarily good rocks for microbial, Dr. Orcutt's work, and vice versa. So there's a little bit of competition on the ship here
yeah.
for good
But the thing
versus
is, we'll
bad
probably be
rocks.
collecting more rocks for dating. So Dr. Ercott or Beth, she wants in particular 2 really good rocks, where for dating we're going to need a lot more because oftentimes, even though it looks really good, it's not quite right. It doesn't quite have the right crystals in there that they need for dating. So we'll be grabbing more of those type of rocks. These
Yeah.
are not gigantic rocks. They're things about fist size to cantaloupe size rocks that
And
we're going to grab.
yes, and from Beth's presentation yesterday, I remember she said it takes a long time to actually study these rocks. So
Yeah,
she will only be able to process like 2 rocks, or she will only collect
yep.
two rocks from each dive.
Yeah,
Right?
that's it, because there's a lot of work involved once the rocks are back up in the laboratory. And if it's not a good rock for the purpose that the geologists and the microbiologists want to use the rock for, then it's a lot of effort for nothing. So again, another reason why we're going to be very, very picky in getting good rocks. And the other thing about the biological samples is that because we are in this spectacular monument, we're just not going to collect any old type of animal. We are looking specifically for things that are new, that are unusual, that are very, very important to the seamount, maybe because they're very, very abundant and we don't recognize them right off the bat. And so we are going to be very, very picky on the animals we collect. And again, we have a lot of collective experience doing this. And we've got the help of a big guide with thousands of images. And we've got wish lists of animals that we're not sure about, we think could be new species. And we're going to use this material to guide us. So yeah, push that up. That's it.
Yo.
We're all getting used to the new technology
Oh,
here.
I was
Yeah, you
talking
want
and
to?
I wasn't realize I wasn't being
Yeah.
heard.
No, go ahead and repeat what you were saying,
Yeah,
Andy.
so I was saying to viewers at home, there is an online guide for the identifying the animals that we're going to be looking at, the sponges and corals. It's the NOAA Office of Exploration and Research Animal Guide. And from that, you can see mostly what we're seeing.
Oh, okay.
We have a little bit more updated guide with some additional animals and critters from other Schmidt Ocean Institute and also Nautilus. But the one you see online, you can identify most of the things that we're saying. So it might be something exciting for the viewers at home to play their own data logger role.
Yeah. So Andy, Dr. Petrancio is here, and he's going to relieve me for dinner while the ROV is still descending, I think. Yeah. Yeah, you're going to relieve me, and I'm going to go down and grab something to eat before we get to the bottom.
All
And
right, enjoy your
so
dinner.
you're going to hear his voice for a little bit, and then I'll be back in a short while.
All right, enjoy dinner, Chris. Chris here is very, very humble, but the truth is that he is has decades and decades of deep sea experience. Originally working with, well, prior to that, but a lot of the time working with the Hawaii Undersea Research Laboratory, which operated a couple of submersibles out of Manoa. They did some of the earliest dive work in Papahanaumokuakea. Before it was even Papahanaumokuakea, it was the northwestern Hawaiian Islands, and they brought some of their submersibles up here and did some really seminal work on understanding what was up here, as well as some of the potential impacts from previous fishing that was active in the monument. and bottom fishing, and also prior to that, also some other deep sea fishing that happened before even the exclusive economic zones went into place. So we're very fortunate to have Dr. Kelly with us here on that to bring his decades and decades of experience to the fore. I'm very excited to be on a cruise with him. Don't often get this amount of time. He was telling me the other day he's such a deep sea aficionado that even though he has retired from the university, he has a sponge lab in his basement at his home in Washington where he studies sponges and documents their incredible structure. One of the coolest things to me about some of these sponges that we're going to look at is that they have this really intricate and complex lattice structure, and it's so unique that they have looked at the way the glass fibers of the sponge intersect and support the walls, and they have studied that to increase the strength of fiber optic cables that we use that run along the bottom of the ocean, because the way that the lattices come together is so strong that it might be better than what we use today to strengthen fiber optic cables. So really interesting what we can learn from these deep sea environments, and it's really, really fascinating. All right, welcome, Emil.
Thanks, Andy. Did Chris treat the audience to his footprints or trails, tracks in the sand analogy for the hotspot seamount trails?
No, not yet.
Yeah, that was one of my favorite parts.
Can you hear me on the mic right now, Dan? Sorry, say that again?
Yes, I can hear you.
Okay.
One of my favorite parts of his brief on the planning for this expedition was the comparison of the hotspot seamount chains to tracks in the sand. We see these various patterns crisscrossing, and the task of geologists is try to determine when they were formed and who formed them, and which hotspots and when, and how was the plate moving. And it's just a really effective analogy, I think.
I agree completely.
Yeah, that's a really interesting way to look at these seamounts, because they form from these hotspots, and then as the plate moves, and I've come to understand the Pacific plate moves about as fast as your fingernail grows.
Yeah, I've heard that one. Some tectonic plates are faster than others.
2.3 inches per year is the roughly fingernail growth speed, and
is it called hotspots because of the marine life, the richness in the marine life, or the potential?
So hotspot is an adjective just for describe an area that's busy in some manner, but in this case, in the geological sense, it's actually a physically hot section of the ocean crust that's heated from below by mantle plumes in many cases, in some cases stretching of the lithosphere due to tension. All sorts of processes underway out there.
Yeah, we learned yesterday from another geologist that we have on board is that for a long time there was debate over whether hotspots and mantle plumes actually exist, or whether it was some other, or it was actually the plate, whether the hotspot stayed stationary or whether the plate moved.
Just so a different spot in the crust.
Yeah, breaks in the crust. And it's only recently that computers have, the processing power of computers has gotten strong enough or fast enough to be able to do models of the mantle of the Earth and actually see these lava or these, magma convective currents that create these hot spots. Essentially, it is a place where the magma is hotter than the surrounding magma, and it actually comes up through the oceanic crust and creates these volcanic activity and volcanic islands. And then ultimately the plate carries it away from the hot spot, and another island is formed, and that's where the footprints in the sand analogy comes from to show us this trajectory of islands and forming from a hotspot.
We see that age progression very obviously in the Hawaiian chain, but we expect to find that through our rock sampling of this much older chain that we'll be exploring.
Yeah, you know what was really fascinating to me was I didn't realize that, so some of these features, we talk about them as being 100 to 120 million years old, but actually those are some of the oldest features on the ocean basin, on the oceanic crust, because on the edges of the plate of the oceanic crust, when the crust gets to that point, then it either goes under a continental shelf and melts back into magma. And so that time period when they are on the basin, on the oceanic crust, is only about 120 million years max. So those are some of the oldest features on the oceanic crust, and I didn't know that at all. The Hawaiian chain's only 80 million years. So the last feature that was generated from the Hawaiian hotspot up in the Emperor Seamounts before it goes underneath the Russian
Aleutian Trench,
Aleutian Trench, or the Kamchatka, is 80 million years old. So the Hawaiian chain's only 80 million years, but the hotspot may have been active for longer than that.
And Andrea Balbas, our supporting scientist, has an interesting idea that the hotspot which created this chain could be related to the Marquesas hotspot, which is still active. And if so, that would be the longest hotspot on Earth, hotspot chain, seamount chain on Earth.
Yeah, I know that. So this might be the longest hotspot chain on the planet if one of the hypotheses is correct about where these seamounts have formed that we're going to be looking at today and over the course of this expedition. And we're just actually outside the boundary of Papahanaumokuakea, just on the border. The Papahanaumokuakea Marine National Monument is 200 miles on either side of the Hawaiian Ridge, or the Hawaiian Seamount Chain, and this is within the exclusive economic zone of the United States, and this seamount is just on the border of that, so we're just outside the boundary of the monument.
Right.
And it's deeper, it's shallower than it's deeper than I thought it was. Was that? I keep forgetting.
Actually, it is shallower. Yeah,
shallower.
The GMRT depth, estimated depth for the seamount based on the satellite radar altimetry is 2,200, 2,300 meters, and we found it to be 1408, I understand. 1,408 meters. But
yes.
We've got a good depth range on this seamount for, hopefully finding some coral and sponge patches.
Yeah, so it just goes to show how we still many things we don't know. The people ask me when we go out on don't we already know about those seamounts? Don't we already know? And we've only mapped a small portion of our oceans, and this is a really good example of the measurements we got from satellite don't match up with what the actual bathymetry measurements from our multi-beam system show us on this site. And many other features are just like this. The satellite gives you a general impression of what the ocean bottom looks like based upon heights, sea level heights, and how gravitational attraction of the mass beneath the sea level, and that's how the satellites estimate what the bottom looks like. But it's quite different than an actual physical ping from a multi-beam bottom profiler like we have on this ship.
David Sandwell of Scripps and Walter Smith from NOAA published that first estimated bathymetry map of the global oceans. And when I saw that, it kind of struck me in the same way that I remember seeing the Marie Tharp, Bruce Heezen map of the world ocean basins in the 1970s when as a school kid. It was just so much more detail. They discovered some new seamount chains that had never been, were uncharted on other maps. And so it's a great cue to where we need more detailed survey from multi-beam.
Yeah, it's incredible how much we still don't know about our oceans, and that's why it's so exciting to be on this ship, to be in this field at this time in our history, to be able to actually physically see these environments, to have the technological capacity to explore them, to map them. And it's just phenomenal watching the multi-beam images come back from the ship live as it's happening. As the ship is transiting over the bottom, we see an actual live image of that bottom coming into focus and doing the multi-beam imagery on a live basis. It's incredible to watch.
The detail we got on the GEOs, the flat-topped or seamounts or table mounts along the Liliuokalani Ridge on the last expedition were just eye-watering, just amazing detail of the ridges coming up to these flat summits.
Yeah.
And then other peaks in that chain, very different, a very bumpy summit. Looks like, multiple volcanic domes on the summit. It's just, it's going to give the geologists, I think, something to sink their teeth into to try to explain how that chain was formed. So hopefully, the plan would be to visit that in April and get some rocks for age dating.
Yeah, and a big part of this expedition is that we are conducting ship-to-shore experiences with classrooms around the country in both English and Olelo, Hawaii, in the Hawaiian language. It's the first time we're ever doing that. We are trying to emphasize the Hawaiian connection with this, the Hawaiian people's connection with this place and the language. And so saying that, I'm going to give you your first vocabulary word for deep sea exploration. So in Hawaii, a mountain is the mauna, M-A-U-N-A, mauna. And kai is the ocean, and seamount is mauna kai. Mauna kai. So that is the seamount in the Hawaiian language in Olelo, Hawaii. So it's an underwater mountain. We'll have more vocabulary coming up for you as our exploration commences. But we're really fortunate to have a wonderful volunteer contingent of Native Hawaiian speakers and fluent in the Hawaiian language to give a name to this expedition, as well as give us the vocabulary for a lot of these features, and also to conduct this live broadcast with the ship, with the schools in Hawaiian language. First time ever we're doing that. It's really exciting.
Great help, yeah. Afrodita, do we have some questions coming in from the Nautilus Live?
The question that keeps recurring is, how deep are we diving?
Right, we'll be going down to 2,700 meters to begin. That depth is selected to try to help define the lower limit of where we may find these dense communities of deep sea corals and sponges. Sweet spot seems to be, based on Chris's experience, 2,400 meters to about 1,800 meters. But so yeah, about 1.5 miles to a mile deep. But we'll see. So we'll start a little bit deeper and work our way up a very interesting ridge towards the summit. And the ridges are excellent places to explore because the currents seem to be stronger there. quite possibly due to internal tides generated, internal waves generated by the tide or internal tides. Or if there are any steady currents flowing around these seamounts, they'll accelerate over those ridges. And so it's a good environment for the filter feeders.
And as we are looking at the monitors, what are the white dust-like particles that we see in the water?
Looking at, we're flowing past, so that's, looks like they're going up, right? But
Yes.
they're actually slowly falling down, but we're descending more quickly. That's marine snow, that's decaying organic matter filtering down from above, and that is the main food source for these benthic creatures, the animals that live on the seafloor. They're dependent on productivity from up above, unless there is some source of nutrients coming from the seafloor, like as is the case for hydrothermal vents or methane seeps.
Yeah, it could also be, I mean, we are about to head into sunset. Even it may also be some of the this greatest migration on Earth of the creatures that are during the evening ascend to close to the surface, and then around sunrise in the morning descend back down. So those are zooplankton and larval forms of some of the animals, the fish and other larval forms. They essentially rise up to near the surface in the early evening and then descend back down again. And it's, yeah, so that might be some part of what we're observing too. Yes.
If you're coming back.
All right, so for anyone listening in, we're currently swapping out some of the teammates so they can go eat dinner. So you will hear some new voices for a couple minutes, but and some quiet time as we're swapping out headsets, swapping out people in the various roles so they can go eat.
Hey, Herc, Emil here. You up on SPL, Trevor?
Yeah, I'm on SPL now.
Hey, just curious on our descent rate.
Descent rate, 30 meters a minute.
30. All right.
We're going to be at the bottom in about 17 minutes.
Very good.
I feel like that went by pretty quickly.
Yeah.
Emily, you may have answered this before I came in to relieve Aphrodita, but how deep is this location that we're going to?
Yes, so we're going down to 2,700 meters. And as our Hercules pilot, Trevor Shepard, just said, we'll be there in about 17 minutes.
How long do we expect this dive to be?
So
How long do we expect the dive to be?
we're planning on 12 hours, but we're ahead of schedule with this earlier than expected dive. The feature wasn't that large after we mapped it, and so we got ahead of the schedule. And so if we need to extend, we can. We've got that flexibility. I would like to see some high density coral and sponge
Yeah,
gardens. And
I think most of our team would.
yeah, but that's a working time for the dive.
Awesome, thank you.
That gives us plenty of time to cover the depth range over which we expect to see these coral and sponge communities. So we'll be working our way up a ridge from a northwest to southeast direction towards the summit. But if we find some interesting communities, we could maybe explore along that depth contour for a little bit, work our way along, sideways along the seamount. Maybe jump to another knoll or something near the ridge.
It's a 1 meg. Yeah. Sure. I did a lot in the red book. You can add those all to the log if you want. There's not really a solved system in that.
Well, I'm not sure if you know this answer, but Steve would have been the right one, except he was just stopping in. Do you know when deep water corals become shallow? Like, what's the depth cutoff between shallow corals and deep water corals?
Well, we have, I think, another good guy to answer that question coming up here. So Chris is back to reassume the watch, and I will gladly defer to him for that answer.
She's eating dinner. I'm swapping her out. Sarah for the win.
Okay.
Yep, there you are.
Let the old guy try again by actually plugging his mic in before he starts to talk. I am back from dinner. This is Chris. And sorry I took quite a while because you can blame the cooks for that. That was pretty darn good food down there.
I
So
agree.
anyway, we are close to the bottom. We're about 2,400 meters, so only 300 meters from the bottom.
Should be just a few minutes. Trevor mentioned it was about 7 minutes or 9 minutes before, so
yeah,
getting close.
10 minutes.
10 minutes.
10 minutes. So Trevor, you're just gonna like run straight on down and ram into the bottom, is that right?
Yeah, first sample is a core sample based on momentum.
You know, what I would advise is maybe you'd sort of pause right above the bottom and sort of take a look and check things out before you ram into the bottom. What do you think?
Yeah, maybe that's a better idea. You're right.
These guys are so professional. They have it all wired exactly how to do all this very, very safely. And
All right, Chris, so my question to Emil, that was, what is the cutoff depth of shallow water corals to deep water corals? Like, when do you differentiate, like, this is a shallow coral, this is a, like, deep living coral? Is there a depth range that cuts that off?
I guess the way that people think about it is you've got, well, I've got to remember my terms here, but you've got corals that are actually deriving a lot of their energy from sunlight, and that's because they have this algae that's in their tissues that's actually photosynthesizing. These are called zooxanthellae, and the algae actually derive and create energy from sunlight, and they pass that energy over to the coral itself. So you have these shallow water corals, and these type of corals generally will go down to maybe, let's say 70 meters or so. It kind of differs on water clarity and temperature and all this other things. And then once you get below that depth, you have a different set of corals. They're usually referred to as Mesophotic corals. And these are corals, some of them are specialized to continue doing a bit of this activity. We got specialized, I think, zooxanthellae. Others are already starting sort of a deeper mode of getting their energy by actually catching prey, small animals or organic debris that's in the water column. And that usually goes down to maybe from 70 to maybe 200 meters or so. And then at that point, the amount of light at that depth is really too low for very many corals to actually utilize photosynthesis. And so once you get down below that depth, then most of the corals are predators essentially, or they're detritivores or things
Okay.
like that.
Oh, that makes sense.
Yeah.
Detritivores. I have not heard that word before. That's cool.
I like it.
Yeah, that's great. Great word.
I mean, sometimes I can be sort of a detritivore depending on what's around the house.
Definitely most people's pet dogs.
Yep.
The corals that we're going to see today are all very, very deep corals, of course. And there's a fair number of people who don't really realize that there are corals down at these depths. In fact, there are amazing corals. A couple of interesting differences is all the shallow water corals, if you look at them, they're all kind of oriented up. You know, you can have these big mounds of, you know, I'll throw out a few scientific names, you know, Porites corals or these branching corals. But the idea is they're trying to expose surface area to sunlight because that's their source of energy. But when you get down below the photic zone, if you will,
yep,
and you're not getting any sunlight, there's no point in really orienting up because a lot of your food is going to come sideways through current. And so you suddenly see this sort of transformation where a lot of these corals suddenly turn into fans and types of things that are going to be filtering horizontally. And that's the type of corals that we're going to see today when we get down there with any luck.
Fingers crossed. But fingers crossed.
Yeah, fingers crossed. I'll bet we're going to see some. Maybe not initially. We're at a depth of 2,700, so we might see a few. But one of the reasons we chose this depth as a start point is we think there's a boundary where we start to see very high density communities that's somewhere between 2,500 and 2,600 meters. So we're going to drop in a little bit below that, and then transect up through there, and we're hoping to see a transformation occur where you go from somewhat barren terrain to very, very lush terrain. That's, of course, it never works out exactly as you hope, but...
Well, we have many hours.
Yep. But what's really exciting, too, is when the ROV gets, you know, just about 10, 20 meters or so off the bottom, and you just start seeing the bottom. If it's a really dense community down there, you can see it even that high up, and you can get really excited.
Yeah. We've seen some on other dives where the corals were bigger than Hercules was, and it was it's crazy. It's
crazy. And those corals are, you know, they're really hard to date. You know, most of them are, but you can expect them to be really, really old. You know, the environment down there is much more stable than it is up near the surface. The temperatures are fairly constant. A lot of times oxygen concentrations and so forth.
Yeah.
So a lot of these animals have a chance to reach very old ages. That was some type of, looked like a little worm there that just went across the screen. There's a lot of these little swimming polychaete worms that you see in the water column, and oftentimes a little distance off of the bottom.
There's a few more now. I must be getting close.
Yeah, and I think as we get closer to the bottom, you'll see even more of this too. Some of these, the swimming worms, they need to take a break every once in a while, so they'll actually go down to the bottom and sit on the bottom and rest for a while, and then off they go again.
Yep.
So I think a lot of them don't want to get too far off. Oh, this is great. We've got Dr. Ken Sulak on the chat room right now, and he is one of the premier fish experts in the world right now, and we were just communicating via email. So thank goodness, because I'm rusty on a lot of my animals here. So
It's Ken, a party over there.
Ken, I'm I'm counting on you to help us out if we see some of these weird deep water fishes.
I mean, I hear all of the fans. Yeah. Those would be much quieter.
All right, 70 meters to go.
The excitement builds.
The excitement builds. No one has ever seen this seamount before. You folks out there watching, you're going to see it for the very first time, just like we are.
The top 3 countries that are watching us right now is the U.S., Canada, and Australia.
All right. Terrific.
There's others, but those are just the top 3.
Yeah.
Can you range that down, please?
What? Oh, they're talking to me.
That's the Doppler. That's fine. You know the Norbit, our down-facing mapping, blah, blah, blah. This is by design, same number from the factory. So it's a fine number. It's just scary and red because it's different, not because it's bad. Yeah, it's different. Absolutely. Yep. Yeah, Meso works. It just needed a squeeze. I don't care. I'm all in favor of trying stuff later. See if it
We're
works.
about 30 meters off the bottom. Another thing that those of you who are new to watching this type of thing, the water isn't blue down there. It's only blue because of our artificial lighting that we use to see. It's absolutely jet black down at these depths. So this is the only way that we can see stuff. But I'll tell you, if we turned off all the lights and we were cruising around down here for a while, you would actually see a lot of phosphorescence and fluorescent animals in the water column down here. So it's not completely devoid of
All right,
light.
we got bottom and sonar here. This is Argus. Argus should be more ranged.
Is that the,
Herc should be less ranged.
are we seeing the bottom already? Oh,
On sonar, yeah.
I
DVLs.
think, yeah, that must be the bottom.
DVLs, yep. 26, 25 meters.
Wow, take a look at it. It looks sedimented, which is a bit of a
We got
surprise.
eyes on in Argus. Argus is 25 meters off bottom. You can see it.
Yeah.
You can hold at 20, and then you can range this down.
Oh, yeah, you
There.
can see it on Herc now, too.
Yeah, look at that.
Okay.
Rush.
That was just the reflection. It's not
Right.
a soft bottom. It's actually pretty hard, rocky bottom. I can see.
Easier to see on Herc's cam.
Yeah. And here we are. We are on bottom.
Yeah.
When we get a chance, Trevor, can we do a white balance once
you
you're all
bet.
sorted?
You betcha.
Thank you.
Aaron, can you
So we're
please
not
swap
seeing
those sonar
tons
head
of
screens?
animals,
I think
but
Herc's on
this
the
is
left.
not surprising
Try.
because we were.
Thank you. I don't know how.
Will I remember how?
Okay.
Okay, you can turn your thrusters on and get your auto heading figured out.
You might have to talk me through it, Trevor. Sorry, it's
I
been a
don't know how
while.
to do it.
You don't know how to do it? Awesome. We're a great team.
Can you click and drag?
Maybe.
That would be neat.
That would be a thing. That would be a thing.
Rip.
Oh.
Meow.
Go over. Ba, ba, ba.
And you can set your heading Antonella to be like 135. Okay. Yep, no big deal.
The first critter I saw, a
Right.
crinoid or a feather star. I was sitting on a rock.
Video, Aaron, full wide on Argus, please. Okay, now you can zoom past the shroud. Thank you.
No problem.
That's all good. And Antonella, you can come up a bit. Let's keep it at 20 to start with. Let's see, what do we got here? It's 30. Thank you. Delta depth of 20. You can give me a DVL reset
Yep.
at your leisure.
Stand by.
Okay, video please full wide on Hercules.
That's full wide.
All right. Still good?
Yes.
All right, resetting. I just have to take a moment to clean this up, and then I'll try the meso again.
Meso? Oh yeah, meso, roger, yeah.
When we collect something, I think. Oh.
Can you turn off the lasers too when we do the balance? Cool, thank you.
Okay, go ahead and zoom there.
Okay.
I'll line it up for you here.
Okay. Other end.
It's probably pretty good.
This Aaron thing's gonna be weird.
I know, it's throwing me off.
I might say video and nav.
There you go.
Can I have, is this all set? Can I do auto XY?
Yes.
We're just gonna lose the feed for a second while it goes. All right, great. Thank you.
If you're done, you can come wide, yeah. Perfect.
Thanks so much. Do you mind if I push in just a tad to get rid of that? Cool.
If I need the extra, I'll ask for full super extra.
Cool, super extra.
Okay, I think we are ready for science.
Okay.
Lasers on, I'm gonna assume, maybe?
Cool.
Well, some of you may have noticed they were doing what's called a white balance exercise, so we needed to be quiet to let them finish this. But we have our first critter that's right down there at the bottom of the screen. This is a sea star. It's, I think, looking at it from this distance, is in the family Brazyngidae. And it's a pretty unusual sea star, although it's a common member of the deep sea here. Why is it unusual? Because most sea stars crawl along the bottom and they scrape stuff off of the rocks and so forth. But this guy, he likes to hold his arms up into the water column and he filters and tries to catch things out of the water.
Video, you can zoom in on the star, please.
Oh, thank you.
Also got a little brittle star next to this sea star. We've got an expert at the Smithsonian, Dr. Chris Ma, who's often watching these dives, and I think he would agree this is probably in the family Brazyngidae. I'm not sure we can go any further than that. Can you go a little bit closer?
Come full wide, please,
arms?
video. I'll get actually physically closer first.
What I'm going to be looking for here is sort of little ridges on sort of the top part of one of the arms, which would help us ID the family that it's in.
Okay, zoom in at your leisure, please. Right there?
A little bit further up toward the disc.
Disc eye?
Yeah, a little bit. Just
I'm working on it. I'm working on it. Oh,
that's okay. I was going to try to use the super duper drawing thing. There we go. Boy.
Get the hang of this.
I'm not seeing these ridges on this. I'm not absolutely sure it's in this Brigidid family. It could have been in, it could be in a
come
sort
wide,
of a
please.
sister family. Thank you very much for that.
Yep.
Maybe a frailty or something like that.
How did you do it?
But great shot.
I didn't.
Thank you.
It's still the same unless magic happened.
I think magic happened.
Did it?
Yeah.
We've got something else there on the upper left. I wonder if we could take a quick look at that.
I have no idea.
Upper left.
Roger.
Kind of leggy white thing.
Leggy white thing.
Yeah, I've got to try this here. Let's see. There's special. There we go.
Yeah, there you go. Nice.
It's a new toy where I can actually draw for the pilots to show them what we're looking at.
You notice how we only put that on the top screen, not on the bottom screen. So you can draw to your heart's content, and we can just ignore it. I'm kidding. Okay, zoom in, please.
I know. The lasers again are 10 centimeters. Oh, it is a coral, our first deep sea coral.
Oh.
And I'm pretty sure this is in a family that's called Primnoidae.
We're really light. We need to pick up some rocks here.
And I am going to guess that it's actually in the genus Norella. And what I'm looking at is, if you look very, very closely, you can see the polyps on it, and they're kind of drooped downward, and that's sort of a characteristic of the genus Norella. These types of corals are very common in deep water.
Let me know when you want me to push in again.
Yeah, the lighting, I'm not happy with the lighting. I got to kind of back away for it to be lit up. You can push in again.
Okay, cool.
Let's see what we look like. I don't know what that yellow thing is on it, but...
All right, you can come down and delta.
I know what the yellow thing
and 20,
is.
I think.
That's actually a predatory mollusk, believe it or not.
Okay, come wide, please.
Yep.
So is our goal to move up this slope?
Yes, please. That would be great.
It looks like it is due east of me.
Okay. We are going to be looking to collect a rock pretty soon. I'm going to be looking for something angular. So if you see something that you think is an appropriate size, it's kind of boxy looking, not too round, then maybe that went up to the, I want to use the machine.
Yeah, tell me, show me with that machine.
Okay, let's see here. I don't know if this one's too big or not.
That one's pretty big.
Okay.
You want me to measure it for you?
Sure, let's put the lasers on it, and this is again one of the great uses of the lasers.
So another great use of the lasers are 10 centimeters apart, which is 4 inches. The manipulators open 4.5 inches wide. So we're looking for about a laser spacing sized rock. So like possibly this one, kind of angular.
Yeah, that's...
The one right...
Is that just one or two? It might be
now.
Two, just sort of stacked one on the other.
Could be, yep.
Oh, there's the lasers. Yeah, that is a little hefty, isn't it?
Yeah, I'm thinking this one right here.
Where the lasers are on?
Yeah, below the one they're on now.
Below the one.
The angular one. This
right
guy
there?
right there.
So that's 10 centimeters.
Ish.
Can
Anyway,
we zoom in on a little?
Go ahead and zoom. Is that the angles you're looking for?
Yeah, well, let's try it. Let's take a look.
Sure. Okay, come full wide, please. Full super extra wide.
Now, before we collect, we're going to do something different here, right?
Yeah.
I wonder if we could sort of just poke it to see if it's loose, and if it is loose and we decide to collect it, then we'll go ahead and do this this chant.
That sounds great. Ow.
Trying to see what that red thing is right above it too. I'm not. It must be another brittle star or something, I would guess, but.
Okay, zoom in, please. Keep all the manip and the brittle star in view. Do you want me to just poke it?
Yeah.
Okay.
Yeah.
Ooh,
And you're seeing exactly why we poke these rocks, because in many cases, even though they look raw, so that's loose.
Yeah, it's solid. That is part of the earth. Wow,
That's
okay.
not bad. It's got a flat side right where the lasers are, I think.
Yeah, I can't get at it though. Okay, well.
Yeah, maybe we'll look around just a little bit more.
Come full wide, please.
Yeah.
Full super extra.